Vaccines have emerged as a transformative component in the broader landscape of cancer treatment, complementing traditional therapies like surgery, chemotherapy, and radiation. Unlike conventional vaccines that prevent infections, cancer vaccines are designed to stimulate the immune system to recognize and combat cancer cells, offering a more targeted and potentially less toxic approach. Cancer vaccines are categorized into two main types, preventative and therapeutic. Preventative vaccines aim to avert cancer development, as seen with the HPV vaccine. Therapeutic vaccines, like the mRNA vaccine, are administered post-diagnosis to treat existing cancers by enhancing the immune system's ability to identify and destroy cancer cells. Today, you will learn more about where Lustgarten has focused its efforts, and thanks to our guests, what is happening in the larger ecosystem of treatment. Our research would not be possible without all of you. Thank you. As always, we encourage you to ask your questions by typing them in the Q&A box below. And it's now my great pleasure to welcome Dr. Andrew Rakeman, Lustgarten's Vice President of Research. Andy, over to you.
Thanks so much, Linda. I'm so excited to be here and talk about this really important and exciting topic, talking about the emergence of pancreatic cancer vaccines and what that's going to mean for the treatment and ultimately the cure of pancreatic cancer in the future. Vaccines, I think, in general, are an extraordinarily exciting topic. I think if we ranked humanity's greatest achievements, the emergence of vaccines against infectious disease is certainly high on that list, if not perhaps topping that list. And vaccines, along with the emergence of antibiotics and improved sanitary conditions, have really transformed human health, absolutely, where previously infectious disease were a #1 cause of death, particularly brutal in children, where at a time before vaccines were present, up to 1 in 5 children would die before the age of 5. Whereas now that's really mostly unheard of, a lot due to the power of vaccines to prevent infectious disease. So after that amazing power of vaccines becoming available, we saw a lot of kind of a calm after the storm, and the new research going into vaccines kind of went into a bit of a post-excitement period. Things sort of slowed down until the time that we're in now, where I think we've seen a real reemergence and on the tip of a renaissance of understanding what can be done with the immune system and with vaccines, not only for infectious disease, but for other diseases as well. And that's due to the emergence of new technologies that allow us to generate vaccines faster and more effectively, discover antigens or the targets that we want to go out after with vaccines in new ways. And importantly, the recognition of the importance of immunotherapies for the treatment of cancers and the real success of some immunotherapies that you've heard about in previous webinars to treat cancers, and kind of the re-recognition that vaccines are a safe and effective way to further boost the immune system against cancers, and pancreatic cancer in particular as being a great target for that. So not only is it a really exciting time to talk about cancer vaccines, in particular pancreatic cancer vaccines, but we have some of the most exciting guests to come in and join me for this discussion. Two of the real leaders in the field who not only have their pulse on what's happening, but are really driving all of the most exciting progress in this field. So I'm going to be joined today by Dr. Neha Zaidi, who's Associate Professor of Oncology at Johns Hopkins Medicine, and Dr. Ben Greenbaum, who is co-director of neoantigen discovery at the Relying Center for Cancer Vaccines at Sloan Kettering. So Dr. Zaidi, Dr. Greenbaum, thank you so much for being here and joining us for this really timely and important conversation. I know that all the supporters and all the folks joining the webinar today are really excited to hear about the progress that's happening in your groups and others that are driving this. Thank you for being here.
So much.
So before we jump into the meat of the conversation and really start to talk about the progress and the promise and the future of vaccines, it'd be great to hear a little bit from each of you about, what it is that you do, and importantly, I think for people to understand, how you got excited about pancreas cancer and about vaccine research and kind of what brought you here today. So if you don't mind, giving a short introduction, talk about what it is that you do, but more importantly, why it is that you do what you do. So maybe Dr. Zaidi, I'll give you the floor first.
Sure. Thank you again, Andy and Linda and the Lustgarten Foundation for having me talk about this exciting topic. So I am a translational researcher at Johns Hopkins. I work at the kind of intersection of the lab and the clinic. So it's been a very exciting time for me to see things kind of move to the clinic and back. I actually specifically got interested in cancer vaccines quite early on as a medical student, actually in New York. And I worked in a basic immunology lab at the time. And my mentor was an immunologist and actually discovered one of the key cells of the immune system, which is the dendritic cell. And the dendritic cell is a very important orchestrator of the immune response. He unfortunately had pancreatic cancer when I was working in the lab. But that's when I first started to work on cancer vaccines and mouse models, actually, of breast cancer and pancreas cancer. And at that time, I had always thought about vaccines in the context of infectious disease, sort of in my pediatric rotations, and how important vaccines were for infectious disease. But the fact that we could actually engage our own immune system and teach our own immune system to recognize and kill cancer cells at that time was such a interesting and innovative concept, particularly as I was going through my oncology rotation, seeing patients on chemotherapy, which is a very important treatment, but these patients had a lot of toxicities, eventually would develop resistance. And one of the key things that I remember really striking me was that the immune system is incredibly complex, but it actually has the potential for memory. So we can get durable responses if we teach the immune system to sort of recognize and kill cancer cells. So that was one of the striking aspects I found of the immune system. And so, it sort of was a full circle when I joined the team at Johns Hopkins to specifically work on pancreatic cancer vaccines. And again, I'm very excited about the translational component because now I actually get to develop and test novel vaccine strategies in the patients in clinical trials. And we're learning so much with the emerging technologies from the clinical trial specimens that we're getting from blood samples, tissue samples. And we can talk about that a little bit later, but that's sort of driving innovation very quickly in this field.
Yeah, thanks, Dr. Zaidi. And I think we'll come back to again and again that importance of the harnessing the power of the body's own immune system, and that evolution has already figured out how to handle a lot of insults, and it's up to us to kind of figure out how to unlock that potential. Well, Dr. Greenbaum, it'd be great to hear from you too about how you came to this field and kind of what inspires and excites you about this.
I actually think there are a lot of parallels in a way. I'm a computational biologist. My background's in physics, and my PhD was in physics. But there were a number of researchers at the time who were interested in applying concepts from physics and computer science and machine learning really to virus evolution. So I switched from physics into biology to really understand how the immune system responds to mostly influenza and HIV infections, why vaccines work in one virus but not another virus, and things like that. And I didn't really think of, I thought of cancer as like surgery, chemotherapy, maybe targeted therapies were at the forefront. And it was this revolution in immunotherapy that really drew me into studying cancer in the first place. So I was really driven by those innovations, which seemed, like an immune checkpoint blockade to be at the same level as antibiotics and vaccines themselves, like real revolutions in using the immune system to make medicines. And then this topic of cancer vaccines was always something that really fascinated me. I was influenced by a colleague at Mount Sinai, where I was before Sloan Kettering, Nina Bardwaj, who I think comes from a similar background as Neha, and to try to see which of these principles could be taken over to the setting of initially for us understanding responses to checkpoint blockade immunotherapies, and then to try to see if it's possible or when it's possible to use vaccines in cancer. So it was, and then I think, you know, like many people, I was really interested specifically to see if any of this could be applied to pancreatic cancer, where, like many people here, I've lost family members, and think of it as a disease where, you know, you really want a lot of different backgrounds and strategies and technologies and points of view to be brought to bear on this important problem. So it was really this, I would say, this immunotherapy revolution that drew me into cancer research. And then pancreatic cancer really is I think a place where you want to try to help as much as you can.
No, thanks, Dr. Greenbaum. And it is, I think, having some success with immunotherapies and having some excitement and still seeing that challenge is definitely something that inspires a lot of the researchers that we work with and talk to. And it's great to hear too, already from the two of your backgrounds topics that, again, we'll come back to, of the need for lots of different approaches and points of view, and, you know, bringing the clinical aspect as well as the research aspect, and, you know, physics and computational biology, and how important it is to be open to all these different approaches here. So maybe before we jump in, it'd be good to take a little bit of a step back and make sure we're all kind of using the same language and talking about things in the same way here. You know, so when we talk about vaccines, and Linda gave a little bit of an overview of different types of vaccines and mRNA versus traditional vaccines. So it'd be a good idea to maybe take a step back and define a little bit of what we mean when we say vaccine. So Dr. Zaidi, if you wouldn't mind, maybe give us a little bit of a overview of, you know, what makes something a vaccine versus a different type of drug, and what different types of vaccines are out there that are being tested today.
Yeah, absolutely. So again, you know, obviously we typically think of vaccines in the context of infectious diseases like the flu vaccine, where we're kind of proactively preparing the body to tackle the infection. But obviously the same concept can be applied to cancer. But there are some differences. So a virus is essentially a foreign entity in the body, but cancer actually develops from normal cells. And what we know about cancer is that there are a series of genetic alterations or mutations that occur in the normal cells that can sometimes convert normal cells into precancer and then eventually into cancer. Now, some of these genetic alterations or mutations can actually lead to the generation of these foreign appearing proteins. Some of these proteins we call neoantigens, and we can discuss that in more detail, because these are actually really good targets for vaccines. The reason being is that these neoantigens, or these proteins, are only expressed on the cancer cells. They're not expressed on normal tissue. So essentially, these are kind of like flags to the immune system, alerting the immune system to try to attack the cancer cells. So the reason why I bring this up is because cancer cells can obviously evade the immune system. They know how to hide from the immune system, but cancer vaccines can actually expose the immune system to these antigens or neoantigens to try to teach the immune system to recognize, engage, and kill the cancer cells. So that's sort of the premise of cancer vaccines. Now, unlike infectious disease vaccines, what we want to see with cancer vaccines is the generation of these key immune cells called the T cells. So the T cells are really the key players in this whole system, in that they can actively engage with the cancer cells and kill the cancer cells. So coming back to your original question, what is a cancer vaccine? I would say a cancer vaccine consists of two key elements. The first element is that we have to deliver that antigen. So this usually is a cancer-specific antigen, a protein that is expressed on the cancer cells, although there can be other types of proteins that are more highly expressed on cancer cells, but more lowly expressed on normal cells. But some of the interest is really in those neoantigen targets that appear more foreign looking to the body. So that's the first component. The second component that we need is what we call an immune adjuvant. So an immune adjuvant is typically something that will enhance the immune system. So we need something to rev up the immune system. We can't just give the antigen. We have to give the second component to really activate the immune system, to get those dendritic cells engaged, which are those key orchestrator cells, and then engage with the T cells that can then go after the cancer cells. So those are kind of the two key ingredients I would say that you need for a cancer vaccine. Now, cancer vaccines can come in many different formats, as you mentioned. Each format kind of has its pros and cons, but there are no real head-to-head trials yet to show us, which format may work for which specific situation or which specific cancer. But just to give you a couple of the key examples of some of the types of cancer vaccines that we're looking at in clinical trials, there are peptide vaccines, there are cell-based vaccines, and then there are mRNA and DNA vaccines. So peptide vaccines have been around for a long time. They essentially consist of short fragments of those proteins that are specific to the cancer, and then they're mixed with this immune adjuvant. So those have been quite safe in clinical trials for many, many years, decades now, and they can generate a good T cell response. Cell-based vaccines are the second type. Those have also been around for a long time. Those are a little bit more challenging to actually manufacture because they're cells, not just proteins. They can come in a couple of different flavors. One of them is that you can actually give irradiated or killed cancer cells. So that would expose the body to a whole host of different cancer antigens. The second type is you can also use a dendritic cell-based cell vaccine. So these are those key dendritic cells that orchestrate the immune cells, and you can expose those dendritic cells to different cancer antigens outside of the body and then inject them into patients as a vaccine. And then the third one is the mRNA or the DNA and the DNA vaccines. These are called nucleic acid-based vaccines. And I would say mRNA vaccines have really surged a lot of excitement, particularly with the work done in pancreas cancer. And after the COVID pandemic, there's been a great deal of interest in the mRNA vaccine. And here you're essentially delivering genetic material that encodes a whole host of antigens. And actually, one of the nice things about this platform is that it can actually co-encode dozens of antigens. And it's a very versatile sort of platform. So that's one very promising strategy as well.
Yeah, so if I'm understanding it right, it really need something that's going to teach the immune system the specificities, that's the antigen, and that could be a protein, a bit of a cell, or some bit of genetic material. And then the adjuvant or some spice that's going to get the immune system excited and make it think that it's getting an infection that's going to want it to ramp up, right?
Yeah,
And-
exactly.
Yeah. And mRNA vaccines, I think that's something people have heard a lot about, both from the excitement of COVID and from the work that you've been involved with, Dr. Greenbaum, where you've taken an approach, and to bring another term in here, of taking a personalized approach, right? And sort of specifically tailoring an approach to each patient. So maybe if you could explain a little bit to the audience here, like what's the difference between a personalized versus an off-the-shelf approach, and why that might be applicable in different situations, or what kind of information you would need to tailor for an individual.
Yeah, I mean, the basic difference is that a personalized vaccine will have at least some of the antigens that are being targeted that Neha mentioned, whether they're neoantigens or others, that are really specific to that person's tumor and are unlikely to appear in someone else's tumor. Usually that's acquired through some kind of genomics approach to assess the specific antigen content of an individual's tumor. Not always precisely, or we're trying to get better at doing that and all of those kinds of things. For example, for the work that we did, a person's pancreatic cancer tumor was surgically resected and then underwent genomic sequencing. And people use that sequencing to estimate what antigens might be best to target in a vaccine. And then a vaccine was manufactured in a very short time scale, about 2 months, more or less, to administer to that specific patient. And of course, there's a lot of advances in biotechnology over the last 20 years that makes that even possible, from next generation genomic sequencing to mRNA vaccine, in our case, design and manufacture. So all of this happens on a backbone of really tremendous advances. Whereas a shared antigen vaccine, and usually personalized vaccines have some shared antigens in them, is targeting antigens or neoantigens that we think are in many, many patients. Or in some cases, even prophylactically, like with the HPV vaccine, where we think they're likely to occur in many patients, or in some of the Hopkins efforts around vaccinating KRAS, for example, which is a common neoantigen target in pancreatic cancer. There are advantages and disadvantages to both approaches. I think if we were able to predict antigens perfectly, and we knew patients definitely had shared antigens, people would like to use shared antigens because the manufacturing part would be a lot easier, and we would be able to move more quickly towards the dream of extending prophylactic vaccination to more people. I think we're still at a moment where we just want to target the best antigens possible. And in some cases, we think that might be a personalized approach, though the field is still, I think, open around any of these questions. Why we would do it in pancreatic cancer is of course a very deep question. Until maybe the last decade or so, a lot of people would have thought that pancreatic cancer was not the best target for this kind of approach. But evidence started to emerge by my colleagues and Neha's colleagues that there was an opportunity possibly in pancreatic cancer for vaccination. And then it also turns out that computationally, pancreatic cancer might, when it is tractable, when it is possible to vaccinate, have a smaller number of possible antigens. And to Neha's point about mRNA vaccines, because they can contain multiple antigens at once, that could be a good intersecting opportunity in pancreatic cancer, where we think there are cases where vaccines are possible. We think there might be a number of antigens available to target that are similar to the amount that you could put in a vaccine. And then that would be a sort of strength for the personalized approach in that case. But I think it's also a very active area of research. And if we ever found really great shared antigens that are reliable and predictable, that would really change things as well.
It's always great for a change to hear about things that make pancreas cancer an optimal target to look at for things, because of that we're so used to hearing about the challenges of pancreas cancer, that the drug resistance, the impermeability of the tumors, the fast moving action. To hear an area where pancreas might be a good first target to look at with these.
It is a really interesting point. There are cancer types, including many that are historically more responsive to classical immunotherapies, where there are just many, many possible antigens. And that makes the prediction problem really hard. And if you have a vaccine platform that can only take in 20 or 30 or 40 antigens, that's a really tough problem. Whereas in pancreas cancer, we think that in some cases, those numbers might be a little bit more manageable.
Right. So maybe now moving on to get to the meat of the discussion and the excitement, and start to talk about what's really the current research and what's exciting, and kind of maybe to give people a preview of the arc. I'll ask you each to start to talk about kind of what it is that's really exciting and inspiring for you today, and we'll talk about what's going on, and we'll close up by asking you for what's on your wish list, or where do you want to be able to go in the future after this. But maybe as a chance to start, so we've kind of laid the groundwork of vaccines are really anything that's going to turn on the immune system in a specific way against what it is that we want to kill, in this case a pancreas cancer cell. Even though there's lots of different ways to do that, they're all sort of going to achieve the same goal, and there's pros and cons and different reasons why you might choose one or the other, but it's really all in the same way. Could be used at different stages of disease. But there's still a lot of work to do. So maybe, I'd let you start with the introduction of Dr. Zaidi. So I'll turn to Dr. Greenbaum to start here to kind of lead off. What's exciting to you right now in your research, or the research that you see going on, or, new technology that you're really seeing changing the landscape?
I mean, I think for us, and this is really due to Dr. Balchandran's efforts that were mentioned, I think we're part of a few efforts where there are some institutions and organizations that are really committing to vaccines as a science that needs to be pursued, rather than something that's done on an occasional basis by some great labs that really believe in the topic.
Mhm.
And because of that, I think that the data collection that's happening around vaccines is starting to happen in a way that's becoming maybe more consistent from setting to setting. And also because of the excitement from a handful of trials, there's a lot more clinical trials happening in this space generally. So the these sort of initial, findings that caused great hope have led to a lot more efforts and a lot more efforts that are being done in a way that's consistent and that intersect with both new technologies in terms of how to sequence and quantify how the immune system is interacting with cancer cells so that the data we're getting is more quantitative, but also starting to intersect with the advances in machine learning and artificial intelligence, which need data that's collected in like a really rational way to shine. And that wasn't the case before because not enough people were working on it, and because some of the technologies like sequencing the cells of our immune system are really only about 14 years old, 15 years old or so. So there, I think it's the biggest excitement is around this kind of moment where there's institutional efforts on the topic, technologies so that we can understand what's happening in successful and unsuccessful trials so that we can do better next time. And a number of investigators and organizations where, really the Lustgarten Foundation has led, and partners in industry who are, taking more shots on goal than they used to pardon what I guess is a pun. So that we're going to get you're going to get more data in a diverse array of settings, and the data that we get out of it is going to be of a higher quality that allows us to be more quantitative going forward.
It's one, not surprising to hear that the emergence of data is something that's going to be transformative, and especially from the computational biologists here, that this is what's exciting you. But let's hear from the translational oncologist perspective, Dr. Zaidi. What's exciting for you, or what do you see in your own work that you think is going to be promising for the future?
Yeah, I mean, I'm excited about a couple of things, and I think Ben put it together quite nicely, and I'll echo some of that. But I think the translational component, we're actually, again, seeing things move in a very iterative process. So we're learning from each step, and we're actually rapidly able to build on that in the following subsequent trial or the subsequent arm. And that's happening very quickly, actually. And I think that in part that it's due to the technologies that we have now. So we are getting, blood specimens, tissue specimens, and we're really able to understand on the most granular single cell level as to what types of immune cells are we actually generating with the vaccine, and what types of immune cells are actually important in protecting patients, for example, from their cancer coming back. And so this sort of very granular single cell information is very important for us to learn what components of a vaccine we actually need to really optimize. So I think the technologies are definitely moving that part forward. We're also getting tissue specimens, so we're able to not only understand at a single cell level what kind of immune cells we're getting, we're able to understand what are they actually recognizing, so we can actually see, are these T cells recognizing the antigen that we're actually delivering. So we're able to really understand the specificity to the most granular level, and we're able to now understand the spatial relationship between the different cell types. So as you know, pancreas cancer is a very complex tumor. We call it a tumor microenvironment in that it has many different cell types that can sometimes block the T cells from getting appropriately activated. So we're able to understand the different signals at play. And that's important because some of the work that was done here by Dr. Liz Jaffe at Johns Hopkins really showed that, a vaccine can induce a T cell, but sometimes you need the right combination of drugs. So I think now we have better immunotherapy agents to actually pair with the vaccine, and those are getting better and better. So that's a very exciting area as well. So I think the technologies, the combination therapies for pancreatic cancer, and what I'm really interested in is the interception part of it. So can we even go after cancer before it develops? Can we intercept precancer? And again, that's something we can maybe touch on later, but that's sort of a goal, I would say, with vaccines.
Yeah, I think we certainly will come back to talking about prevention and all the different stages where vaccines might be applicable. And one thing I think I heard from both of you in your answers that I would say for me is personally the most exciting right now is that all of these things are happening and being tested in clinical trials in actual patients, and even in some cases seeing some benefit for patients in these early stage trials, and even looking at the questions that are starting to come through on Q&A, which encourage people to continue to put them in. I see people asking about clinical trials, about experiences their family members have had in clinical trials. So maybe Dr. Zaidi, could you give just a overview of what the landscape of clinical trials for pancreas cancer vaccines looks like right now? Kind of what types of vaccines are being studied, what stages of disease and combinations that you alluded to, and where that's possible, are we seeing results from these yet?
Yeah, absolutely. So most of the trials, I would say, are kind of in the earlier stage. They are kind of moving towards, larger studies, but some of the very early studies do show some very promising signals in terms of clinical efficacy. There are two, I guess, buckets of trials, which I think Ben alluded to already, but they're the personalized vaccines where you develop a vaccine that's very specific to that individual patient's tumor. And then there are what we call the off-the-shelf approaches. So these are vaccines typically against mutations in KRAS, which is one of the first driver mutations that occurs in pancreas cancer and actually occurs in up to 90% of pancreas cancers. So it is a kind of a high frequency target that we can use for vaccines, and it covers the majority of pancreas cancer patients. So those are also being tested in clinical trials. So we have both the personalized and the off-the-shelf KRAS vaccine trials that are currently underway. Most of the trials are currently in the minimal residual disease setting. And what I mean by that is these patients have undergone surgery and gotten their chemotherapy and then go on to receive the vaccine. So unfortunately, as many of you know, patients who are eligible for surgery and get the standard chemotherapy, there's still a 50 to 70% chance of that cancer coming back. So these strategies are employed to reduce the chances of the cancer coming back. And I think one of the lessons we've sort of learned over the decades is that vaccines tend to work better in clinical settings where there's less tumor. So, when we're thinking about minimally residual disease, there could be some cells after chemotherapy that we can't see on scan, but that's when a vaccine tends to be more effective. When there are large tumors, when there's metastatic disease, vaccines have many more hurdles to overcome. So there's a lot more immunosuppressive signals in the cancer. There's a lot more heterogeneity, so differences in the types of cancer cells and in the types of antigens. So there are more barriers to overcome in that situation. So some of these recent kind of earlier stage clinical trials have really focused in the adjuvant setting, meaning after surgery and chemotherapy. And that's where we're actually seeing a lot more successes, not only in pancreas cancer, but other cancers like melanoma, where there's an mRNA vaccine that showed a clinical efficacy signal in a randomized trial. So that was very exciting. So those studies are currently ongoing. They're now expanding to be randomized trials and larger trials. Another interesting clinical setting is also the neoadjuvant space. So this is before patients go to surgery. And so there are going to be trials looking at the vaccine in that situation where the cancer hasn't spread yet. It's still localized to the pancreas. But this could also be an opportunity for us to vaccinate before surgery to try to improve overall outcomes. But it also helps us understand when we vaccinate before surgery, what is actually happening at the tissue level? What other signals are at play? What other signals do we need to bypass? And so that's some of the work that had been done here at Hopkins, really building on what we call a platform trial, where we understand the signals, then we build on that by adding different agents that then try to circumvent those immunosuppressive signals. So right now, I think those are kind of the most promising spaces, the adjuvant and neoadjuvant. There are a couple of trials in the metastatic space. Usually we give them with immune checkpoint inhibitors. Even in the adjuvant setting, some of these trials are testing vaccines plus immune checkpoint inhibitors. And we think that you probably need both, because T cells tend to get functionally exhausted over time, and that word is literal in the sense that they tend to lose their efficacy, so they tend to be less activated. So you need that second signal with the immune checkpoint inhibitor to maintain activation, to maintain the quality of the T cell response over time.
So really there's work happening in the clinic across the spectrum of stages of disease, from prevention, which we'll talk about later, to people at early disease or after surgery, and even some late stage. But you really think that the most promise is going to be when we can catch when there's low disease burden, right? Either after surgery or very early. And you also, coming back, keep bringing up this importance of learning from patients and learning from how people respond. And now, so turning to you, Dr. Greenbaum, in the work that you've been doing with Dr. Balachandran that you referenced, testing these personalized mRNA vaccines after surgery, you've also had some time to follow up on these patients and learn about who responds, who doesn't respond, and what comes from there. So maybe if you could give a little bit of a flavor for the audience here about what you're learning from these trials and how that's influencing thoughts about how to further optimize your approach in the future.
Yeah, I mean, we're very fortunate now that these trials are happening in the setting of a new vaccine center, the Land Center for Cancer Vaccines, and it has a computational sort of unit that's funded by the Tau Foundation, just to try to make sure that even, as I was saying before, even in trials which are maybe don't meet their endpoints, that we still extract data in a uniform way. In terms of the first trial, one of the main things that we were able to do was to try to understand quantitatively the dynamics of the T cell response to the vaccines that Neha was referring to, the cells that we think are kind of recognizing and killing the tumor cells at the end of the day. And one approach that we've done, which we called clone track, is to really densely sequence the immune response in a patient's blood while they're getting vaccinated, so that we could label the T cells that we think were involved in the vaccine response, try to validate that, but then also try to continue to sequence the immune repertoire of those patients over long periods of time, so that we could understand the durability of the response. Does it seem to be waning? How does that correlate with things like recurrence of a tumor? And also to simultaneously do something called single cell T cell sequencing, so that we understand the behavior of those responding T cells even more deeply. And that's all in collaboration with Dr. Balachandran. There's definitely a theme of making sure that we get the most out of precious patient data that's attached to all of this. As I think we talked about a while ago, a lot of this started because we and others were collecting a lot of data from unusual responders to traditional treatment of pancreatic cancer and trying to understand why someone who would seem respond to standard treatment many decades later, seemed to be a real outlier in terms of how well their response was going, and then measuring those patients in a way that we could see that it seemed like the immune system had a role in that response. So, the ability to leverage patient data was a big part of how we got here in the first place, particularly at institutions like MSK or Hopkins, where there's enough volume that you would see unusual responders in both directions and try to be quantitative about why that was happening. And a lot of those efforts were actually funded by the Lustgarten Foundation. So that's sort of how we're trying to use quantitative approaches to sort of understand the dynamics of response. And then as we get more patients, we'll be able to bring in some more advanced computational approaches to try to predict those sorts of dynamics even earlier in the vaccination cycle, or try to even use that information to improve upon them going forward.
So again, going back to this importance of collecting data, engagement and involvement of the patient, and participation in clinical trials really being key. That, I think a big part of the reason why we've had the progress that we've had so far is because of the trials that came before, and keeping up data, whether it's the work that you've done or Dr.
Yeah, and
the work at the platform studies at Hopkins that have really led to these iterative improvements in combination therapies, including vaccines.
a lot of observations from those earlier efforts that maybe, to other investigators, maybe didn't look like ideal, but making sure we got the most information from even those cases, and then people were like, hey, but this small subset of times we think something happens that might give us a general lesson about how we can move forward. It's so important, as is the important, I think, of interdisciplinary teams that are really led by domain experts in pancreas cancer, which is, I think, a model that both of us believe in. Those things really, you can go a long way, I think.
I agree entirely. And maybe it's a bit of a transition here to talk about, again, areas where I think we need lots of different technologies and lots of different data and understanding and disciplines coming together is to address the area of prevention, which I know is something that you've really been a leader in, Dr. Zaidi, and pushing us forward and looking at the questions, lots of questions coming through about, vaccines for prevention for people with family history, with known mutations, with, cysts or IPMNs or other sorts of pre-malignant lesions in the pancreas, or after having surgery and have been, in remission or in no measurable disease for a long period of time. So maybe you could talk a little bit about kind of what's different about vaccines for prevention, both in terms of the vaccine design, what it takes to bring them into the clinic, which is also different, talking about treating people who are healthy versus treating people who are facing a deadly disease. And kind of why that's so exciting right now, and what else is really needed to bring that to the next level.
Yeah, no, thank you. Obviously, this is a topic I'm very excited about, and this is something that I think is high risk, high reward, and thanks to the Lustgarten Foundation for really, funding this to move this forward. But this is an area that we've been working on at Johns Hopkins to develop vaccines for the interception of pancreas cancer. And it turns out that pancreas cancer may actually be an ideal cancer for cancer interception for a couple of different reasons. First is, I mentioned this KRAS mutation actually exists in the majority of precursor lesions to pancreas cancer. And this has gotten to be a more complex area, lots of work going on in the precancer space, very exciting with the new technologies we have that can actually study the three-dimensionality of these precursor lesions, which are actually microscopic. You have to look at them under the microscope to see them. But these KRAS mutations occur very early on, so are really interesting antigen target to go after in the interception space. The second thing we know, and we've been studying this at Johns Hopkins, is that when you look at the immune cells related to precancer, they're actually activated T cells that exist that are around these precancers. But as the precancer develops into cancer, more of those immunosuppressive signals come into play and shut down the T cells. The T cells appear more exhausted or more suppressive. So the T cells actually may be more prime for vaccination and for education at the precancerous stage. And so now, again, with the technologies we have, we're really starting to understand the immune milieu around those pancreas cancer precursor lesions. So we have actually developed a peptide vaccine here at Johns Hopkins that targets the most common KRAS mutations. And we've tested this already in 20 individuals who are at risk of developing pancreas cancer. We are now in our second phase of the study where we're also vaccinating individuals with pancreatic cysts, which actually are visible on imaging. And those individuals who unfortunately have to go to surgery because the risk of transformation to cancer is so high, we're actually vaccinating them prior to surgery to help us understand, are we generating the right immune cells? Are these immune cells getting to the place we want them to be, which is the precancer? So we're in the midst of that second stage of our study, which will hopefully help us understand what these immune cells are actually doing in the precancerous space. But it's a really exciting area, especially for pancreas cancer, because unfortunately, the majority of patients get diagnosed when the disease is advanced. Even when it is surgically resected, the recurrence rate is high. And now we actually know that up to 10% of all newly diagnosed pancreas cancer do have some inherited predisposition. So now every single patient who comes here gets genetic testing. So that not only has implications therapeutically for them, but also allows their family members to understand their risk. And there are now actually screening programs around the country, some of which are also funded by Lustgarten, to screen these individuals and to actually pick up the cancer early. But right now, even those patients who are at risk, the only cure is surgery, which is pretty tough. So we are looking at developing these interception strategies before cancer develops.
So maybe just to follow up on that a little bit. So you have the initial trial that you did in people at high risk, and you could look at immune response and see some positive results there. You're now looking in the IPMN patients, which is very exciting because you'll get to see at the tissue level what's happening. What would it take to bring that out into something that now really gets it to the next step where you could start to think about becoming available more broadly in the clinic, or something that could be used overall? And how is that different from, developing a new therapy to treat people who have an existing tumor?
Yeah, I think it's a little bit different because, these patients may or may, hopefully will not develop pancreas cancer, but have a risk of developing pancreas cancer. So first, obviously, the threshold of establishing safety is different when we go into individuals who don't have cancer. So we had to, in our initial trial, safety was one of the primary endpoints. And obviously this vaccine did show a very good safety signal. So that's the first thing we look at. The second thing, as you mentioned, is, are we generating T cell responses? And then the third item is, in our IPMN trial, we'll see, are these T cells are actually being activated? Are they recognizing the precancer cells, and can they kill the precancer cells? I think this fundamental knowledge will allow us to hopefully broaden the trial to be more inclusive to maybe patients who are at slightly less risk, but who are still in these high-risk screening programs. But obviously, the dream and the goal is to, be able to give such a vaccine to, anyone who is at risk of pancreas cancer, because it does have such broad coverage.
Thank you for that, Dr. Zaidi. And I hope that addressed a number of the questions that I saw coming up, rather than taking them each one by one. But one thing, just to clarify a little bit, when we talk about being at high risk for pancreas cancer, it's still a relatively low risk overall, but it's higher than the general population. And that's either because you have one or more family members who have had pancreas cancer previously, you have one of the few mutations that you inherit from a parent. These are like BRCA mutations and a few others that give higher risk for pancreas cancer, or you have a known cyst or IPMN that can be detected that looks troubling. That would be get followed, correct? Are there any other reasons that would put somebody in a high risk category?
Yes, yeah, I think you're right. And I apologize for not clarifying. High risk
Yeah.
is still relatively low risk, but it's just what we call kind of the, quantify as the highest risk. But yeah, I think it's just the certain genetic mutations that we screen for, certain IPMN lesions. Not all of them go on, obviously, to develop cancer, but there's certain characteristics that are more troublesome.
So the time goes fast when there's lots of exciting science to talk about, and we are coming up on the hour. And I do want to promise I'm going to give you each a chance to give your wish list for the future of where we want to be. One other question that came up in the chat that I want to address here. So you've heard a lot of work that's funded by Lustgarten Foundation. You've heard about work that's funded by others as well. Work that's supported by the Lustgarten Foundation is all supported by our generous donors and supporters that come in. We are at a time right now where there are changes to the overall landscape for research funding, with expected changes to federal funding to come, a lot of different kinds of discussion around vaccines and the way vaccines specifically would be supported and looked at, especially for infectious disease vaccines. But I do want to assure everyone on the phone here that this is the commitment of the Lustgarten Foundation to continue to support the most important and the most impactful pancreatic cancer research is unwavering. And it is something that we will continue to find ways to drive the most promising research forward, regardless of what happens in the outside world, but are always looking to make sure that we're in a position to take advantage of the most promising research and fill the gaps that are being left behind by others in support there. So the commitment from Lustgarten is steadfast and unwavering, and will remain committed to learning from the best science, learning from our patients to do better for the patients in the future. So with that, I'll turn it over to, we'll start with you, Dr. Greenbaum. If anything you want to close with, or kind of give your what it is that you're looking forward to seeing come out in the future, or what you see as the biggest roadblock or obstacles to be able to progress this research forward as fast as our patients need it.
I mean, first, thank you for your support and for doing this. I think that these kind of advanced interdisciplinary efforts on big scientific questions, and this is definitely a big scientific question, have always benefited from the support of philanthropy, even when those outside things you're talking about weren't as threatening or seemed different. So I think it really, this kind of work doesn't happen without that. That's sort of at the leading edge and is bringing in the brightest minds. And I think that's one of the things that I'm most optimistic about, is that the level of talent from a lot of different fields that we've been able to attract to this question over the last, say, 3 or 4 years is just fundamentally different. People are excited about this. They want to help. They're going to overcome roadblocks and setbacks and challenges that are going to happen. Not everything is going to look perfect. But you're starting to see a critical mass of really talented investigators, and that's the most exciting thing. It's also the most fragile thing, if we can't support them. And so there is this kind of, I think, theme that we certainly, the two of us, think something really exciting can happen if we all get together and put our best foot forward and make sure the best talent in America and internationally can work on it. But that it's happening at the same time where we're also concerned about the long-term future of being able to do exactly that. So I think that's really the thing that I'm always the most excited about, which is that in addition to all of the infrastructure that we're able to bring to this question, just the excitement and amazing cross-disciplinary talent that really wants to work on this at a national level, and work together with other investigators, build up a great program at MSK, work with people at Hopkins, and try to move the ball forward. So I think when you see sort of, young, brilliant people spontaneously send you emails and show up at your doorstep and, really say that I want to do this instead of a fellowship in computer science at Stanford or something, that's when you know that we need to capitalize on this to at least make sure that we know more and can help more patients, you know, today than we did yesterday.
Yeah, it's fantastic. So I'm hearing we need data, we need talent, we need dollars, and it's great to hear the talent. And I think the two of you are very representative of this. The kind of passion and that people bring to this is inspiring.
The kind of, I just think that the amount and interdisciplinary nature and just incredible talent of young investigators that just in an unsolicited way want to work on this is something we really all need to take advantage of, because if nothing else, we'll be better off from that experience.
Agree, bro. Dr. Zaidi, how about you? What's on your wish list for a brighter future for your patients?
Yeah, I mean, I think Ben touched upon it all. I will add that I think we obviously, thanks to the support of Lustgarten and many other, sources of funding, I think we can't stop now, no matter what, because we are seeing the improvement in clinic, and that is what's really exciting to me. The fact that patients are, responding to treatment, they're tolerating treatment so much better. We're getting better outcomes. After decades of work on cancer vaccines, I think right now is a critical moment in, bringing everyone together, as Ben already mentioned. But I think this concept of team science is going to continue to go a long way, including nurturing the next generation, but bringing together, within our institution, inter-institution, you're working with industry, for example, who are conducting many of these clinical trials, integrating data sets, using machine learning. Those are the types of things that I think will advance the field even faster and allow us to build on our current vaccine strategies.
Excellent. Thank you. So it sounds like although we still have our work cut out for us, we still have a lot to do. We have momentum on our side. We have talent on our side. And I think can definitely say that from the Lustgarten perspective, we are not going to stop. I know from both of your perspectives, you're not going to stop, and we will do our best to make sure that we take advantage of all of the great work that you've done and the great work that is to be done in the future. So thank you so much for joining us today for this. I know it's been informative for our participants, and we look forward to hearing the next updates on your work in the future. Linda, I'll turn it back to you.
Thanks, Andy, and thank you, Dr. Greenbaum, and thank you, Dr. Zaidi. This conversation is so important. It really reminds our community, who has invested so much for so long into our work, that there are results of the work and that those results are happening right now, and that the science all these years has been iterative. We just heard Dr. Greenbaum talk about how the work that they're doing is based on the years of the data that they were able to then maximize for this type of work. So the fact that our community has been so patient all these years and continue to invest in the research has been that's what really inspires me, and I'm sure that it's inspiring to all the researchers that we support, and of course it's inspiring to our community. So thank you again, everyone. Beginning in September, we look forward to welcoming everyone back for the fall season of Lustgarten Live. Some of the highlights include unlocking early detection from precancerous clues to AI-driven discovery. And we're thrilled that we have something new, the Lustgarten Sprint to Discovery. It'll be a few short talks from postdocs in our Lustgarten labs. And there will be a co-presentation with Northwell Health Cancer Institute entitled Delivering Point of Care, Genomic and Genetic Testing for Pancreatic Cancer Patients. Thank you again, everyone, for joining us today. It was a great conversation, and have a great summer, everyone. Thank you.